What I'd like to do today is go through a little bit of the background and the history of both GemPharmatech and some of the brief history of genetically engineered mouse models. So as many of you may know that virtually 90% of all Nobel Prizes that have been awarded in physiology and medicine have involved animal models. And that is one of the most exciting underpinnings and the need for better research models to enable research discoveries as well as better pharmaceutical development. This really hit the forefront in the late 80s with the development of embryonic stem cell technology and the ability to really genetically engineer mice beyond random transgenics, which started really in the late 70s. With this advent of ESL technology, around 2005, Francis Collins, who was then the director of the Human Genome Research Institute, and had recently published the first draft of both the human and mouse genome, assembled a team in Cold Spring Harbor to discuss what could be done with better mouse models, because many were being made in the research community. They were not standardized, and in fact, they were difficult to acquire. Many people would spend a year or two developing mouse models with ESLs, and then basically would continue to work with them and not necessarily make them available to the general public. So with this meeting called the Banbury Conference, it was decided that it would be worthwhile for the US, with assistance from other interested parties, primarily in Europe at this time, to develop an entire library of embryonic stem cells where each gene that encodes a protein in the mouse genome would be knocked out, and this would then be made available to the entire research community, both public and private. The goal then is that these would then be developed into mice. There would be some phenotyping analysis, and then this data also would be provided to the entire public. However, at this time, the funding really only covered making a library of the embryonic stem cells, and I was quite happy to be involved in the Knockout Mouse project, both at its inception and also managing the first 5 years of that program in combination with my colleagues at NIH, particularly Colin Fletcher. 5 years after the initiation of that, we were able to get additional funding of over $120 million from the US NIH group to develop the International Mouse Phenotyping Consortium, which really was kicked off by the MRC Harwell Group and the Sanger Institute as well as the foundational members of this phenotyping consortium. I was very honored to be selected as the first executive director of the IMPC. And with that, we recruited a number of different groups, which eventually totaled 16 different research institutes around the world in 13 different countries. And so this network would then take the embryonic stem cells and create mice and provide a phenotyping analysis of them, which is in place into a database and then presented to the entire scientific community. You were also able to order the live mice from the various members of the IMPC who had created those mice, again, enabling research throughout the world. This is very exciting, and I think it was going quite well. And then in 2012, the world changed, and the world changed with the CRISPR craze. We went from having this entire collection of embryonic stem cells, which were primarily knockouts, which had a reporter gene in them, to the ability to create knockouts or other mutations in actual zygotes in the mouse embryo. This saved nearly 6 months of time in creating the mouse models, and also despite the great utility of embryonic stem cells, virtually half of them did not go germline, so we could not make a mouse. So the cost was tremendous. The advent of the CRISPR technology really allowed us to cut the cost by a factor of about 2/3. So with that, we moved from the embryonic stem cell, and the IMPC started making mice directly with these knockouts. Unfortunately, at that stage, the technology was still difficult and expensive to do things such as conditional knockouts or inserting reporter genes, so we created a library of knockout mice with the IMPC. In parallel with this, GemPharmatech was a spin out from the MARC, the Mouse Animal Research Center in Nanjing, which was a member of the IMPC. And the MARC group, which later spun out GemPharmatech, started advancing CRISPR technology to do model customization services, adding conditional knockouts, humanization, creating immunodeficient mice, and developing preclinical efficacy and testing, as well as mouse breeding services. So in essence, what has been able to be accomplished at GemPharmatech is really that initial dream that we had in 2005, which is to make any modification in the mouse genome at will and provide better mouse models for better pharmaceutical development. And what we will do today is focus in on one of these aspects, and that's really the conditional knockout mouse program. But GemPharmatech as an organization offers all of the different services and products that you see on this. And our goal in the US, as I mentioned, is to expand this to continue to create better mouse models for human disease, better mouse models for better pharmaceutical development. And again, we believe that these technologies have really allowed us, to create amazing tools to accelerate research and discovery. And I'm very excited, having worked in this field for more than 25 years, to see how quickly and rapidly it has grown. And I'm excited and really believe that in the next 20 years, we're going to see amazing breakthrough therapeutics that's unparalleled because of these better mouse models to create, again, better pharmaceuticals.